primary productivity is the rate at which photosynthesis occurs. Nutrients such as nitrogen and phosphorus are needed for photosynthesis to take place. However, nutrients in excess can be harmful, leading to algal blooms, eutrophication, hypoxia, and even dead zones. This led us to our questions. Does nutrient enrichment increase marine primary productivity in the St. Augustine Marina? As well as, is there a difference in which nutrient is most significant in increasing or decreasing primary productivity? In order to investigate our research questions, we decided on a light and dark bottle experiment. The first step of our process was measuring out the standards based on the molecular weights of the nutrients, and then we calculated what amount to add to get our desired concentrations.
In order to get our initial measurements, we needed to record the salinity using a refractometer and use that measurement as well as the barometric pressure to calibrate the YSI Pro Solo dissolved oxygen probe. Once the probe was calibrated, we took triplicate measurements of dissolved oxygen to allow for a more precise reading.
To prep our bottles, we needed to thoroughly rinse them with the Marina water before filling them to minimize any chance of cross-contamination from prior use. After the bottles were rinsed, we filled them up to the top, placing the cap on underwater to avoid getting any air bubbles. Before adding our standards to the treatment groups, we removed exactly 250 microliters of water using a pipette so that our precise measurements of nutrient standards did not overflow once the cap was placed. We had 3 light and 3 dark bottles for each treatment. Each nutrient has a specified amount according to calculations stated previously. The St. Augustine Marina is a nitrogen-limited system, therefore the nutrients that we used were nitrogen-based as we hypothesized there would be a greater effect. We used ammonium chloride, potassium nitrate, and urea. Each of the bottles were thoroughly shaken after the nutrients were added. The dark bottles were then wrapped in aluminum foil, which helped keep the bottles from heating up. Bottles were then placed into bags according to groups and threaded through the rope secured on the dock. One dye bag was then secured to the dock as well, ensuring that if the rope came loose, one experiment remained. Once all 4 dye bags were placed in the water, the rope was secured to the dock by a cleat hitch knot. The bags were left for 24 hours to incubate.
24 hours later.
After the 24 hours of incubation, the dye bags were collected, ropes picked up, and delivered back to the lab. Once back to the lab, bottles were taken out and placed according to groups on the table. Bottles were mixed again before being placed on the stir plate. A magnet was then added to the bottles and the plate was turned up to approximately 500 RPMs. The DO probe was placed on top of the bottle and the final measurements of dissolved oxygen and temperature were taken once the meter stabilized. This was repeated with every group. Light bottles measured first, then dark bottles. The light bottles contained both photosynthesis and respiration reactions. The dark bottles tell us just the rate of respiration because the dark bottles inhibit sunlight from getting in and therefore prevent the photosynthesis from occurring. By subtracting the dark bottle from the light bottle, we are able to look at overall primary productivity. After measurements, bottles were dumped and then rinsed 3 times with deionized water to prepare for the next experiment.